next trick, I shall conjure up the 21st amino acid, selenocysteine. What? I know I told you there were only like 20 common amino acids, but this is the 21st. It's called selenocysteine. It's just like cysteine, except it has a selenium instead of sulfur. And this is going to give it important functions and allow it to serve as like an help protein serve as like antioxidant function, so helping protect our cells from like free radicals. We'll talk more about what this means. So there's only about 30 proteins in our bodies that actually use selenocysteine, so it's not that common, but it is super important. In order to get this selenocysteine into proteins, your cells have to use some really cool trickery that involves switching like a stop codon, so the instructions for telling the ribosome making the protein to that you've reached the end of the protein. The selenocysteine basically takes over the stop codon, and there's this special element in the RNA when for these proteins that use selenocysteine that kind of like tricks the machinery into, before the release factor can come and like clip off the chain, it's going to sneak in and like use that stop codon as its own codon. So codon is just like the instructions for putting in an amino acid, like a 3-letter code. We'll get into all of this, but anyway, so it's really important, and so we're going to talk about basically how it does this, which is this cool trickery, as well as how the differences between selenium and sulfur and oxygen can help explain how all of this goes down and why this is so cool. So selenocysteine should remind you of cysteine, which should remind you of serine. So these are all 3 amino acids that have this similar structure, but they have different versions of this like something H group. And so in serine, you have an OH, so you have an oxygen. With cysteine, you have an SH, so you have a sulfur. And with selenocysteine, you have a selenium, so you have a this SEH, so you have a selenium. And so when we have an OH, we call that a hydroxyl or an alcohol group. When we have an SH, we have a sulfhydryl or a thiol group. And then when we have a SEH, we have a selenyl group. And we'll get into why these similarities and differences between them. But for now, just know that, so these are below each other on the periodic table. So they're going to have the same number of valence electrons, more on this in a minute. And basically, the key thing for now is that selenium is going to be easier to deprotonate to form this like selenate anion, and the bonds that form to it are weaker. So selenium is going to be, you're growing bigger as you go down. And these electrons, so the things that are going to allow them to react and form bonds, are going to be more loosely held. You can think of kind of like a giant hand trying to shake like a tiny hand. It's going to have a harder like time forming strong bonds. So we're going to be able to like swap out things bound to selenium more easily. One of the places we can see this is by helping like reverse damage caused by reactive oxygen species. So we'll get more into this, but basically, as we saw when we talked about cysteine, it can get oxidized to form these like cross links, and sometimes you don't want these cross links, and so you need a way to reverse them. And selenothioate is going to help reverse it back to cysteine, so it can act as an antioxidant again. So it can do this because a bond to this like mixed selenium sulfur bond is going to be weaker than this sulfur bond. And so then this is easier to break than that was. And so this is going to allow you to break up those bonds. And we're also going to see how it can serve to prevent that damage from happening in the first place through this like glutathione peroxidase. So we'll get more into that. But in order to do those functions, this selenocysteine isn't just like floating around doing this on its own. It's going to be doing this in the context of an enzyme. So there's a bigger overall protein, and then the selenocysteine is just like sticking out in part of it to do this reaction. And so in order to get it into the enzyme, in order to make it as part of the protein, you're going to need to actually like get the protein making machinery to put it in the chain as it's making the protein. Proteins are just like long chains of amino acids that fold up to form a functional protein, and how they fold and function is going to be dictated by what amino acids they have, because these all have these different properties, as we've been talking about over the past 20 days. What order those amino acids are put in is going to depend on the instructions, the genetic instructions. And so you have the gene, which is like the permanent DNA copy of the instructions. Then there you make like this messenger RNA intermediate that the ribosome then reads into proteins. And so the process of translation is where this ribosome, this protein making complex, is going to travel along the messenger RNA. So that RNA copy of the instructions is going to travel along, and it's going to a tRNA, so a transfer RNA, is going to bring the corresponding amino acid that corresponds to the codon. So there's like 3-letter RNA word that spells for that amino acid. And so the ribosome is this big complex. It's made up of protein and RNA, mostly RNA. And what's going to happen is that the ribosome is going to be traveling along that messenger RNA, so along that RNA copy of the protein instructions. And it's going to be reading it codon by codon. So it goes like 3-letter steps. And so each of those steps is going to be a codon. And each of these codons is going to basically have, say, put in this amino acid. And then how it works is that the tRNA with the match has like this transfer RNA has a matching anticodon. So like a complementary anticodon. And then that's going to bind, and it's going to bring in the corresponding amino acid to get added to the chain. This is going to keep going and keep going. So some of these things called like elongation factors are going to help this happen, because basically what you need to spend a little energy to get this to push over. So the ribosome has these like 3 sites. You're going to have, when the incoming tRNA comes, the chain is going to get transferred to the incoming amino acid. So the growing chain is going to get transferred to the incoming amino acid, and that incoming amino acid is going to be coming into like this A spot, and then this is being held in the P spot. And so now you have to shift things over to the P spot, and then shift that guy to the E spot, the exit, so we can leave. So basically you just have to spend you have these elongation factors help things out. And then finally, what's going to happen is that this is going to keep going until a stop codon is reached. And then when that stop codon is reached, what's going to happen is is that a release factor is going to bind, and then this is going to get cut off. And so this release factor, instead of being a tRNA, it's a little protein. And what it's going to do is it's going to bind, and it's going to cause this to hydrolyze. So this chain is going to get broken. And so these release factors are going to bind when you have one of these stop codons show up. So one of these like UAA, UAG, or UGA. But in the case of selenocysteine, it's actually going to take over this UGA. So when the UGA shows up in the stop codon site, it's going to, this tRNA for selenocysteine is actually going to bind instead, and it's going to bring the selenocysteine to be incorporated. How this works is that it's only going to happen if in like certain genes, and it's only going to happen because it has this special like element, this selenocysteine incorporation sequence. When we're talking about the messenger RNA, we are focused on this like protein making instructions part. But then past the protein making instructions, there's actually a little bit of regulatory information. We call this like the 3' untranslated region, or the 3' UTR. And so this region isn't translated, it doesn't have, because translating is when you put in the protein letters, and so there's no instructions for protein letters here. It's just regulatory information. And we call it the 3' because basically we're talking about reading this RNA from like this 5' to the 3'. So basically it just refers to the directionality of the RNA letters. So in the 3' UTR of selenoproteins, you have this special secondary structure in the RNA. And so RNA can kind of like fold up into like hairpins and stuff like this. And there's this special structure in the mRNA of these select selenoproteins. And so this is going to allow them to use this stop codon put in selenocysteine instead of a stop codon. But this is only going to happen if you have that special element. So in all the rest of the cases, this UGA will tell the stop, and the release factor will bind instead. But why you need this, how this works basically is this structure is going to hold all of this stuff nearby. So there's also this special elongation factor that's needed for this selenocysteine. And so this is going to bind to the tRNA and bind to this these like helper protein and stuff, and to this RNA structure. And so basically all of this is going to be hanging out like right nearby, so that when that stop codon shows up, it can sneak in there before that release factor can bind. So the release factor could still bind here, but this is going to bind more tightly and it's already there. So basically it doesn't have to, the cell needs to find that release factor. So the release factor has to like come in and bind. And so when this is right there, it can sneak in before that release factor can bind. And this will allow it to be used instead of using this as a stop codon. And then the real stop codon would be down here somewhere in between this like selenocysteine incorporation spot and the SECCIS element. But in order to do this, of course, you need to get this selenocysteine made. And so how it's made is that it's actually made based from serine. So basically what happens is that when your body is normally like making amino acids, or when it's normally getting amino acids onto the tRNA to be added, how it works is a couple steps. And so you have the amino acid activation step. And so basically here you're spending ATP to make this like energetically favorable and make this so that it can be like attacking group by the tRNA. And so this is going to more in ATP and other posts, but basically it's kind of like a coiled, like a clamp spring. So you have these negatively charged phosphate groups and they're held really close together. If you break them apart, then you release energy that you can use to do things. And you make this bond more reactive, because when you break this, then you're making this happier too. So we call these like high energy bonds because they have like a high potential like chemical energy. But this is going to make this bond more vulnerable basically. And so it'll be vulnerable. It can be attacked by this tRNA. So the tRNA has this like OH group sticking up here, and it can then attack the adenylated amino acid. So we call this, so this is ATP. When this amino acid gets activated, you're going to have this AMP that binds. So like this is like an adenosine and adenosine monophosphate. And so when you have this like adenylated amino acid and then it binds, the tRNA is going to take that amino acid and break this this bond to the AMP, which is going to get released. So you're investing some energy, but it's worth it because you're assuring that the right amino acid is loaded, because the ribosome is not going to care what's there. The ribosome is only going to care about the anti-codon matching. So you want to make sure that you put the right amino acid onto the tRNA. So how are you going to do this with selenocysteine, however? So selenocysteine, it does this in a tricky way. So it has its own tRNA. So it has a tRNA that's going to match the stop codon. So it's not going to use it's serine's tRNA, but it is going to use serine's tRNA synthetase. So basically, this is going to be the thing that's going to add the amino acid onto the tRNA in that activation step. And so it's going to use the same, this takes like a synthetase to help with this process. And selenocysteine is going to use serine's tRNA synthetase. And so it's actually going to get loaded with a serine. And then it's going to get modified after you put the serine on here. So the serine is going to get loaded onto this tRNA for selenocysteine. Then what's going to happen is that we need to replace this OH with an SEH. And so this is going to be done in a couple steps. So basically you have this there's this kinase, so this basically you have this kinase, so this like phosphate group adder, and it's going to add the phosphate to this OH. So now we have phosphorylated serine. So we talked about how we could like phosphorylate as a post-translational modification, serine could get phosphorylated. So in that case, we're talking about like after this was put into the chain, then in that case, we're talking about like after serine is put into the chain, it can get modified by this phosphorylation. But here we're making these modifications like pre-translational. So this is all like before we even put the amino acid into the chain, and we're going to remove that phosphate. But by making one of these higher energy, easier to break bonds, we're going to make this more attacky. And so we can swap out the selenium. And when we swap out the selenium, then we're able to sneak it. We have the selenium charged tRNA that we can then use to sneak it into proteins. So the swapping out is going to be done with this enzyme called O-phosphoserol tRNA sec selenocysteine tRNA sync synthetase. Yeah, so we abbreviate it SEPSEC-S. And so this is going to be doing the swapping, but it has to be done. It's a little trickier than just like swapping it out because you need this selenium to be made basically like fresh. So when you have, your body doesn't want to keep like a big stock of free selenium around because for reasons we'll get into, it's more reactive and it can cause problems if you have just like a bunch of free stuff floating around. So instead of having this like free floating selenium, what's going to happen is that your body is going to basically store it in the form of proteins. So we talked about how like having selenium in proteins is pretty rare. But there's actually and like selenium containing proteins, so like selenoproteins, typically have just like a single selenocysteine residue. So residue is basically when you link up amino acids, we call like the remainders residue. So once amino acids are linked, we can refer to what was an amino acid now as a residue. But we often use the terms interchangeably, amino acid and residue. But basically most selenoproteins have like one selenocysteine. But this one protein, selenoprotein P, SLP, it has like 10 of these. And so it's really good at transporting selenium safely and kind of like safekeeping it. So it's home to about like 40 to 50% of the selenium in your blood plasma. So like the cell part of your blood is held by this SLP protein. So basically you have it stored as selenocysteine in this protein form. This can get broken down by selenocysteine lyase, and this is going to give you Se2-, which selenophosphate synthetase, SBS2, is going to combine with ATP to make selenophosphate as needed. So basically, this is going to get added in this like form of phosphorus this form of phosphate that's connected to a selenium. So yeah, so it's kind of complicated, but basically now it can get snuck into proteins. So I showed you some stuff on PDB 101 before. It's this really great website from the PDB, the Protein Databank. And they have a good article on like selenocysteine synthetase. So this is like the special elongation factor that's going to help. It's going to be like help sneaking it in. You can see where it like binds RNA and stuff. This is selenocysteine synthetase and selenophosphate synthetase. So you can see the structure of some of these cool enzymes, the reaction helper uppers that are going to help this all happen. And so I encourage you to explore this. So we actually have two amino acids that have sulfur. So we have methionine and cysteine. So you can have methionine as well as selenocysteine, and we take advantage of this in the lab sometimes when we're doing like crystallography. If you give like the growing cells that you're trying to get to make your protein, if you give them selenomethionine instead of methionine, they can incorporate that. And then you have, this is this can help you with crystallography because it's like a heavier atom, and it gives you this like signal that you can use to help with this like phasing problem, which I'm not going to go into, but it's a helpful tool. But typically, but our bodies, we're not actually like using selenomethionine, but we are using selenocysteine. So because we can get selenomethionine like in our food and stuff, we can sometimes like accidentally misincorporate it, but that's not really that big of a, if you just like accidentally had a selenomethionine in your protein, it wouldn't be a selenoprotein. But it's a selenocysteine containing protein, that is a selenoprotein, because we're telling the cells like to put in the selenocysteine. And so why are we doing that? So we saw when we were talking about the differences between cysteine and serine that there are some benefits of having a sulfur versus a versus an oxygen in certain cases. For example, we saw how with the cysteine we could deprotonate it more easily, and we could form these weaker bonds, and we could form like cross links and stuff. So let's talk about where these differences are coming from, because basically the differences, you're kind of going to this through this trend when you go from oxygen to sulfur to selenium. The things we said about sulfur versus oxygen, we can now say like selenium versus sulfur versus oxygen. So the trends are just going to keep going. We're going to keep getting like weaker bonds, more nucleophilic, less electronegative. We'll get more into what these talk. Let's get into these terms and talk about them in more detail. When we're talking about oxygen, sulfur, selenium, we're talking about elements, and elements come in like units of atoms. And so basically these elements are defined by the number of protons. So atoms are made up of smaller parts called protons, neutrons, and electrons. The protons are positively charged and the neutrons are neutral. These hang out in this like dense central core called the nucleus. Then there's this cloud of electrons hanging out, whizzing around them, which is negatively charged. Elements are defined by the number of protons. So the number of protons is going to be fixed for a given element, but the number of electrons can change. And basically, it's not this simplistic, but we can draw these elements as, we can draw atoms as kind of having their electrons in these shells. And in this shell model, basically the electrons on the outermost electrons, we call these valence electrons. And for reasons outside the scope of this post, atoms are going to want to have a full outer shell of these electrons. And they can get this full outer shell by forming bonds. So they can share a single pair of electrons for a single bond, or two pairs for a double bond. And so this is how they can fulfill their shells. If you go down the periodic table, you have the same number of these valence electrons. So they'll need the same number of electrons to fill that shell. And so if you go down the periodic table, these elements that are in the same column, these elements that are in the same column or group, these are going to have similar properties because they're going to need to undergo the same type of reactions in order to fill their shell. However, as you go down the periodic table, what's happening is that you're adding a shell of electrons in between them. So you have the same number on the outside, but you have a whole shell inside. And so your atoms are getting bigger, and those valence electrons are getting farther out. When they're farther out, they're farther from the pole of the positively charged protons in the nucleus. So I like to think of it kind of as atoms as kind of like dog walkers, and these electrons as dogs. And so the outermost dogs are the valence electrons, and they're the highest energy. They're the furthest away from the pole. And so they are the most willing to like be reactive and that sort of thing. If they feel the pole less, they're going to be less what we call electronegative. So electronegativity is basically like how tight a pole does an atom have on its electrons. So we can have these trends. So if you have more protons, so as you go across the periodic table, the elements are differing by a proton. If you add more protons, you have like a stronger dog walker, a stronger leash. You have more of that positive pull that can be pulling on those electrons. But as you go down, you're adding like a shell of electrons in between. You're going to get this like shielding, and you're going to have the valence electrons farther away, and they're going to feel like the pull less. So they're going to be more reactive. And so if we look at oxygen versus sulfur and selenate versus selenium, we can see that we're getting bigger. These valence electrons are getting further away. So we're getting less electronegative. We're having a loose, not as tight of a hold over those electrons. So they're going to be more reactive, and they're going to form weaker bonds because you get less like overlap and stuff between when you're sharing electrons, there's like less overlap between your clouds and stuff. So we can talk, there's some different terms that we can use, like basicity and nucleophilicity. The most common way we talk about like a base is something that steals a proton. And we can also talk about, so base is a special case of like acting as a nucleophile. So a nucleophile is something that has more electrons or more like electron density than it wants. And so in order to get some help sharing those electrons, it's going to attack something that wants electrons, so an electrophile, and then they can like form a bond to it. And we can get like substitution reactions and that sort of thing. If a nucleophile instead grabs a proton, then we say it like acted as a base and gets protonated. So one thing that can get kind of confusing when we're talking about like oxygen versus sulfur versus selenium is like solvent effects. So what's happening is when you have a bigger element and we're in this like watery environment, the water is going to like be kind of shielding and like kind of caging in our nucleophile. So the bigger you are, the more resistant kind of you are to getting trapped. So when you have a sulfur, when you have a selenium, you're going to have a bigger atom, which is going to be able to better withstand the this like caging effect. So although the O- is going to be less happy than this S-, because you have more concentration of charge with this O- than you have with the S-, the S- is going to be less trapped. And so it's going to be more nucleophilic. And so this means that it's going to be more willing to act as a nucleophile and like attack something, and attack an electrophile. However, because that OH because that O- is so much more unhappy, it's going to be much less likely to form in the first place, basically. And so it's going to be a much stronger base. It's going to be harder to deprotonate it. And so when we look at these, we'll see that serine has a pKa of about 13. So much more on this in other posts, but basically the pKa is the point at which half will be protonated and half will be deprotonated. If you go to a higher pH, then you have fewer protons available. So you're more likely to be deprotonated. If you go to a lower pH than the pKa, you have more protons available, and so you're more likely to be protonated. So you have to compare the pKa to the pH in order to determine like how much of it's going to be protonated. So if you have a high pKa, like you have with an alcohol or a hydroxyl group, then you're basically always going to be at a lower pH than the pKa, and so you're basically always going to be protonated. In our bodies, we have a pH of around like 7.4. So we're well below the pKa of the hydroxyl. And so it's basically going to be really hard to deprotonate this. If you have a thiol, so if you have like a sulfhydryl group like we see with cysteine, now you have a lower pKa. You have a lower pKa because this thiolate, so because the conjugate base of this acid is more stable than the hydroxylate, because it's bigger, it can spread out that charge more easily. Now you have a lower pKa. It's still above the pH of the body, so most of the sulfur, most of the cysteine in your cells is going to be in this SH form, this protonated form, But you will see some in this deprotonated form, especially in like the active site of enzymes and stuff, where you can have other amino acids helping deprotonate it. When you look at selenocysteine, now your pKa is actually even below the pH of the body, because the selenate form is a lot more common than you would see with the deprotonated form of either of these guys. Because it's even bigger, this large size is going to let it spread out the charge. So this is happier, but it's also most nucleophilic because of the effects of that water. The water is caging this hydroxylate, and this is going to be less trapped by water, and so it's more going to be able to attack better as a nucleophile. So it's easiest to deprotonate, most nucleophilic, so it can react. It's basically like more reactive, even though it's basically like more reactive, even though it's happier than these guys, because it can react. Like it's not really happy. It still has, because it has all these, it has a negative charge. Things don't really like being negatively charged that much. And so it'll still react as a nucleophile, and a stronger nucleophile than these guys, because it's not trapped. So it can act as a nucleophile. And so this is going to be and when it attacks as a nucleophile, it's able to form bonds. But because the large size is also going to make the bonds that it forms weaker. And so this is going to allow you to kind of like swap things out when you form a bond to a selenium. And so this is why we're able to like break up the sulfur-sulfur bonds with sulfur-selenium bonds. But because they're easier to break, you wouldn't want this to be like, you wouldn't want to use this for making like bonds that you wanted to be super sturdy and super permanent, but you can use it to like as like a sensor, kind of to see like the oxidative environment, because it can get oxidized and reduced and that sort of thing, as well as to form these intermediates like these selenosulfide bonds that can then get broken. And so one of the key selenoproteins in our body is this family of enzymes called glutathione peroxidases. And their function is basically to neutralize the threat posed by hydrogen peroxide. It's going to take this hydrogen peroxide and form water. And this is going to protect our cells. Why is this going to protect our cells? So hydrogen peroxide, this oxygen-oxygen bond is going to be unstable. So oxygen is really electronegative, so it has a tight pull over its electrons. But when you have two oxygens together, they're both kind of like pulling each other apart. And so this bond can break. And when it breaks, it can do this in this way, this like homolytic cleavage, where each one of these is going to keep an electron. So it's going to have this reactive radical, basically, a radical is where you have an unpaired electron. And when you have a radical, you can get these like chain reactions of radicals where one thing is attacking another thing, and basically you don't want that to happen. And so glutathione peroxidases, they're going to be able to turn this into water to neutralize the threat from it before it can form those radicals. And so there are important functions of like hydrogen peroxide and stuff, but you need to be able to tightly control the levels, and you don't want excess. So glutathione peroxidase can play a really important role in this, in reducing this. So what do I mean by reducing this? Well, we need to talk about oxidation and reduction. So when we're thinking about like atoms sharing pairs of electrons to form bonds, this is basically they're overlapping kind of like parts of their electron clouds. And when they're sharing electrons, they might not share fairly. So as we saw with like electronegative things, they would pull more on one than the other. And so basically we have this thing called, like when we talk about like oxidation, it's kind of like if you were to break these up, who would the electrons go with? And so if something is bonded to an oxygen, which is something, if something's bonded to something that's really electronegative, like an oxygen, then the electrons that are in the shared electrons are actually going to be hanging out more with like the oxygen than they would with the hydrogen. And so therefore, if something forms more bonds to oxygen, or they form bonds to something that's more electronegative than themselves, it doesn't have to be oxygen, then you are going to be oxidized. You're basically losing electrons because the oxygen is kind of taking some of the electrons in the shared bond. And with reduction, you have a gain of electrons or electron density. And so basically, if you lose a bond to oxygen and instead form like a bond to hydrogen, hydrogen doesn't have a very tight pull over its electrons. And so you can pull its electrons towards you. So you'd be more electronegative than it is. And so you get more of the electrons. And so we call that reduction. And these are going to go side by side. So if you oxidize one thing, you have to reduce another. When we talked about cysteine, we talked about how it can get oxidized. So it can get oxidized to like sulfenic acid and sulfinic acid and sulfonic acid. And you can see we're forming more and more bonds to oxygen. Oxygen is going to be pulling those electrons away. So we're losing electron density. We're becoming oxidized. And you could also become oxidized when you form these disulfide bonds, these cross-links, because you're losing bonds to hydrogen and forming bonds to sulfur, which is more electronegative than the hydrogen, even though it's not as electronegative as the oxygen. When we looked at our trends of electronegative, we saw that if we go to the right, we're getting more electronegative because we're adding protons. But we have the same, so we're basically with oxygen, sulfur, and selenium, we're in the same amount of rightness. But the further you go away, you have the shielding effect. So even though you're adding more protons as you go down, that is the effect of those protons is like basically lost because you have all of this shielding from the inner shells, and the electrons on the outside the valence electrons are going to be farther away. And so they're not going to feel the pull of all those extra protons you just added. And so as we go down a column, we're going to get more we're going to get less electronegative. And so the oxygen is going to be more of these three. Oxygen would be the most electronegative. Selenium is going to be the least electronegative. So this means that if a selenium forms a bond to oxygen, it'll be oxidized. But also if selenium forms a bond to sulfur, it'll be oxidized. And if sulfur forms a bond to selenium, it'll be reduced. So we can have these redox reactions occurring between like sulfur and selenium that aren't even involving oxygen. One of the key molecules we're going to see help us in this reaction is this molecule called glutathione, which is basically formed from glutamate, cysteine, and glycine. And it can go between these reduced and oxidized forms that actually involve this cross-link between these sulfur atoms. Our cells can use glutathione in its reduced form. They kind of like keep a stock of it that they can be using to help reduce proteins. And it's going to help us regenerate our glutathione peroxidase after we use the glutathione peroxidase, after we oxidize the glutathione peroxidase, or in order to neutralize the hydrogen peroxide. Because remember, our goal is to get the hydrogen peroxide, not to do this, not to go that radical way. So instead, we want to break it up. We want to neutralize this threat safely, converting it into water. But if you look at this, you'll see that you have two oxygens here, and water has one oxygen. So clearly we need to do something out. We can't just convert this straight into water. What's going to happen is we're actually going to oxidize this glutathione peroxidase. We're actually going to put this OH onto selenium. And so this is going to then, so when we're oxidizing this glutathione peroxidase, now we need to reduce this in order to reset the system. And so how this is going to be done is that you're first going to reduce it by forming a bond to sulfur. And so when we saw sulfur form a bond to sulfur, that was an oxidation, right? But that was because we were replacing a bond to hydrogen to a bond to sulfur, and sulfur have the same electronegativity, but sulfur is less electronegative than oxygen. So if we swap out a bond to sulfur for a bond to an oxygen, well, now we've reduced this. But now we have the selenium sulfur bond. So now we want to get it back to selenium to hydrogen, which would be another reduction, because now you're going a bond from sulfur to a bond to hydrogen. And so hydrogen is less electronegative, so this is going to be another reduction, and so we need another reducing equivalent of this glutathione. And so what's going to happen is this glutathione is going to be oxidized by forming another sulfur-sulfur bond to this copy of glutathione that you have stuck on GPX. And the reason why you're able to do this so like easily is that these selenium sulfur bonds, these selenium sulfide bonds are going to be weaker. Remember, the bonds to selenium are weaker and easier to break. And so this is going to be a lot easier than if you were trying to do it from like a cysteine cross-link. And so in fact, when our bodies use this like thioredoxin, this other little protein that's going to help break up cysteine crosslinks, in order to like reset everything, it's going to like thioredoxin reductases, they're going to use selenium. And so you have selenium showing up in these various places where you're going to be resetting systems based on the ability of this selenium to act as a really good nucleophile, and the ability, but also the ability of these bonds that it forms to sulfur to break more easily. So selenium itself wasn't discovered until 1817, and this was by the Swedish chemist Jan Jacob Berzelius. So it's a pretty famous chemist, and he found it in a sulfuric acid factory. And he named it selenium after the Greek moon goddess Selene. And selenocysteine itself isn't essential in the dietary sense, meaning that our bodies make it ourselves, but the selenium part is. And so selenocysteine is the only amino acid that requires an essential dietary micronutrient, so selenium. And so we get the selenium through foods. So in the plant form and stuff, we get it from plants, we get it as selenomethionine, and then like from animal proteins, we get it as selenocysteine. But either of these can get broken down in our body so that we can make selenocysteine. And so we don't actually use selenomethionine really. It can kind of sometimes get like misincorporated, but it's not really thought to cause problems. But we don't have like genetic instructions saying like put in a selenomethionine here or whatever. So really we're just talking about getting selenium from these foods, but then we're transforming it to the forms that we want and using it as selenocysteine, and incorporating it into proteins under very specific conditions. So selenocysteine, we didn't even know that it was incorporated into proteins, however, until the 1970s. The late biochemist Theresa Stratman, who is a really amazing woman, and much of what we know about selenium and selenocysteine and selenoproteins is thanks to her.